If you take a medication and it doesn't work, are your gut bacteria to blame? Hi, and welcome to Microbial Minutes, the American Society for Microbiology's update on what's hot in the microbial sciences. I'm Madeline Barron, Science Communication Specialist at ASM, and today we are talking about how the gut microbiota, the community of microbes living in the intestine, influences the activity and efficacy of pharmaceutical drugs. We will also touch on how scientists are developing digital tools to study these complex drug microbiota interactions to inform personalized treatment regimens. Now, people take medications for many reasons, from relieving pain to managing diabetes or depression, among countless others. But the thing is, not every drug works the same for every person, if at all. And why is that? Well, bodies are complex and there isn't a single reason. However, it is becoming increasingly clear that the gut microbiota may have something to do with it. In fact, researchers have found that the bacteria in the gut can regulate how drugs work as well as their side effects. These interactions between gut microbes and pharmaceutical drugs are bidirectional. That is, drugs can alter gut microbiota composition and function, which has implications for how the drugs are then metabolized and used by the body. There are a wide variety of drugs with known ties to the microbiota. Shown here is a small selection of them. How different drugs alter the microbiota varies from one drug to the next, though impacts can include a reduction in microbial diversity and increased growth of certain potentially pathogenic bacterial species. The ways in which medications alter the microbiota are largely indirect. They somehow alter aspects of the gut environment, such as its nutrient profile, to influence bacterial growth. However, the effects may also be direct, as exemplified by a key study in which researchers screened upwards of 1,000 marketed drugs, including antipsychotic and anti-inflammatory medications, against 40 representative strains of human gut bacteria. They showed that 24% of the drugs could kill at least one of the strains in vitro, with anti-neoplastics, hormones, and compounds that target the nervous system, inhibiting gut bacteria more than other medications. These results hinted that the bactericidal capabilities of human-targeting drugs could perhaps shape microbiome composition in vivo. In any case, the types of bacteria living in the gut at a given time has very real ramifications for an individual's response to a drug. For example, in a study published a few years ago, researchers found that metformin, a drug used to treat type 2 diabetes, had strong effects on the composition of the microbiota in human patients. When they transformed the metformin-altered microbiota from those patients into germ-free mice, that is, those without a microbiota of their own, via fecal transplants, the mice showed improved glucose tolerance. Specifically, the animals had lower blood plasma glucose concentrations compared to those that received microbiota from human patients prior to metformin treatment. The findings suggest that microbes may modulate some of metformin's anti-diabetic effects. In another recent study, researchers showed in 2 independent human cohorts that heterogeneity in responses to statins, which are drugs used to lower cholesterol, was tied to the microbiota. That is, an enrichment of species belonging to the Bacteroides genus, which is a prevalent component of the human gut microbial community, and reduced microbiota diversity were associated with intense on-target responses to statins. They were also associated with adverse effects, such as disrupted control of blood glucose levels. The mechanisms through which the microbiota modulates the response to statins are still unclear. However, in some cases, bacteria are known to directly metabolize medications to determine where they go and what they do in the body. Levodopa, a drug used to treat Parkinson's disease, is a well-known example. In order for levodopa to work, it must cross the blood-brain barrier and be metabolized by a human enzyme in the brain to form the neurotransmitter dopamine, the active therapeutic agent. Nevertheless, certain gut bacteria, along with peripheral human enzymes, can metabolize the drug before it crosses the blood-brain barrier, likely reducing its availability and efficacy. This means that someone's microbiota could inform how much of the drug gets to where it needs to be, and, ultimately, how well it works. And while there is a compound called carbidopa that is administered alongside levodopa to block peripheral metabolism of the drug, it's a host-targeting inhibitor and does not appear to be active against the microbiota, thus is not entirely effective. So why is all of this important? Well, understanding how an individual's microbiome influences their response to a drug could help determine when it is prescribed. This fits in the broader framework of precision or personalized medicine. That is, when medical decisions are tailored to a specific patient population based on genetic, environmental, lifestyle, and increasingly microbiome factors. Advancements toward precision medical practices depend on technological innovations for analyzing these complex intersecting health factors. To that end, a study published in Nature Biotechnology earlier this year provided a step in the right direction. In this study, researchers created genome-scale metabolic reconstructions, essentially digital models, of 7,302 human-associated microbial strains representing over 1,700 species. The database, called Agora2, summarizes knowledge of the biochemical and physiological traits of microbes, particularly with regards to drug metabolism. It can be used to model strain-resolved drug degradation and biotransformation capabilities of microbial communities. So how exactly does Agora2 work? The researchers tested it out using metagenomic data from a previous study, including a cohort of patients with colorectal cancer and healthy controls. They mapped the data to the Agora2 database, then used computational modeling to predict the drug metabolizing functions of those microbiomes. What they found is that 97% of named microbial strains in the data could be mapped to Agora2, highlighting the broad microbial scope and coverage of the resource. Moreover, there was significant heterogeneity in drug metabolizing potential of the microbiomes. For example, only 53% of the microbiomes examined were predicted to metabolize digoxin, a drug used to treat heart failure. The researchers also noted that not all microbiomes equally activated basalazide, a drug used to treat inflammatory bowel disease, suggesting not every individual may benefit from the drug. This result is particularly interesting given another recent study in which gut microbiome metagenomics, metatranscriptomics, and metabolomics obtained from patients with IBD were used to uncover 12 microbial enzymes associated with inactivation of 5-ASA, the therapeutic byproduct of basalazide and other related IBD drugs, thus adding some potential mechanistic insights into these observations. Finally, age, sex, disease state, and body mass index were also associated with microbiome drug metabolism, pointing to the intersection between host and microbial factors in drug responses. So these findings highlight the utility of tools like Agora2 for teasing apart if and how gut microbial communities metabolize pharmaceutical drugs. The study's authors suggest that such digital stratification of patients into responders and non-responders for a given drug could then be validated in clinical trials and ultimately inform treatments. For instance, an individual's microbiome composition, along with other factors, may be used to determine whether they are given drug A or drug B. It may also be possible to alter the microbiome composition of patients to influence how they respond to a drug, or perhaps design and administer compounds alongside drugs to protect them from microbial manipulation, similar to how carbidopa protects levodopa from peripheral host metabolism. And it's worth noting that the application of digital tools for understanding microbial drug interactions extends beyond human targeting medications and gut bacteria. Computational resources are also proving useful for developing novel antibiotics to purposely kill bacteria in an age of growing antimicrobial resistance. Essentially, as technology advances, so does our ability to learn about and manage the microbes we interact with. And so what are the key takeaways here? Well, the activity and efficacy of pharmaceutical drugs are influenced by interactions with the gut microbiota. Understanding these interactions is key for advancing microbiome-informed precision medicine. And ultimately, gaining this knowledge requires a growing repertoire of computational tools that help us make sense of the complex metabolisms and interactions of microbial communities. And that's all for today. Don't forget to subscribe to ASM's YouTube channel and ring the bell to be notified whenever a new Microbial Minutes drops. I'm Madeline Barron. I'd like to thank you for listening and thank Ray Ortega for production, and I'll talk to you next time.